Hi, so I'm going to be talking about in vivo and ex vivo gene therapies and specifically about their efficacy, their challenges, and the ethical implications of such technology. So, just a brief introduction. Sorry, my computer seems to not. There we go. Just a brief introduction to who I am. So my name is Wendy. My mentor is Luke Riggin. And I became really interested in this topic because of my biotechnology and AP Bio class. And I am based in Moraga, which is in the Bay Area of Northern California. All right, so a quick overview of what we're going to be looking at today. So first, we're going to be talking about ex vivo gene therapy, so the lentiviral gene therapy and adipocyte LV, as well as in vivo therapy. And then we're going to move on into the ethics of this technology. So here we go, ex vivo gene therapy. Now, if you're not familiar with this topic, ex vivo gene therapy is where you remove the patient's cells and you modify them outside the body before reinserting it into the patient. And the benefits of this gene therapy is that there's no need for donors, which decreases the risk of rejection when it comes to receiving these therapies, as they are tailored to the patient themselves. And some successful applications recently include adenosine deaminase deficiency or ADASCID. And also, this is just like a autologous hematopoietic stem cell treatment from bone marrow taken from the patient, which is then modified with a specific gene before reinsertion. It's been proven to work, and they did studies in UCLA over this. And as for unresectable or metastatic melanoma, so cancer, this is where ex vivo is done after failure from a PD-1 blocker, which is a different type of treatment. And it seemed to work better than the PD-1 blocker. All right. And for lentiviral gene therapy, so a lentivirus is derived from the HIV virus, and it is a retrovirus. So there is a single strand of RNA within it. And it contains three genes, so the envelope, the gag, and the pole. And when you are modifying this gene therapy, you do have to separate these three in order to ensure a safe integration into the host genome. And when it comes to lentiviral delivery, again, it has to be stripped of pathogens and is given the viral payload, so whatever you desire to be given to the patient, and then it is injected into the patient. And again, these viral payloads can be anything that you want. So CRISPR, therapeutic genes, anything that you can think of putting into a patient. And some applications include a BECMA. Now, this is an FDA approved treatment. It's for recurring myeloma following two or more different types of treatment. And Kymriah is a refractory B cell precursor for acute lymphoblastic leukemia. So both are different cancer treatments. And now we're going to go into adipocytes. So an adipocyte is a fat cell, and we are interested in genetically modified adipocytes or GMAX. So we want to use GMAX for diabetes and familial alkat deficiency, which is an issue with cholesterol in this processing. So once this therapy is administered, it integrates itself into the fat cells, and the great thing about it when it comes to its applications is that it can be used for diabetes, again with the familial alkat deficiency, and as well as lipodystrophy. And adipocytes, when you use them, they are great in that they spread out very evenly throughout the body in the area that you want to target, so that they're very effective that way. All right, and now we're going to go into in vivo therapy. So in vivo gene therapy is different from ex vivo gene therapy in that it is the direct delivery of the therapy. So instead of taking cells from the patient's body, like in ex vivo, and then modifying it and putting it back in vivo gene therapy is usually delivered through different means like shots or injections and IVs. So the benefits of this is that it directly targets the tissue and the issues that they have, and it is widely applicable. So it's not tailored for one person like it is for ex vivo. It can be given to a large amount of people. And some applications currently are for hemophilia and cancer immunotherapy. All right. For adeno-associated virus, so AAV is a viral vector, and it can be transduced into both dividing and non-dividing cells, which means that it can be put into brain cells, which are non-dividing, or muscle cells, which are dividing, giving it a more versatile use. And the issues, however, with this is that it is a very small payload, which means that it can't carry a lot of material, and the results do vary, so it's very inconsistent. As for applications, Becvez is a FDA-approved medication, and it's for those with hemophilia B who are more undergoing different, well, trying to simplify this, it's more for people who are going under hemophilia B treatment that have not worked yet, and they have a history of hemorrhages. And in terms of liquid nanoparticle, so LNP is shorthand. It's made of four lipids. So you have the ionizable cationic lipids, the polyethylene glycoconjugated lipids, helper lipids, and phospholipids. So when you think of helper lipids, you're thinking of cholesterol. So it increases the cell interaction with the LNP. And then for phospholipids, it's just making stability for the vector. Now, the benefits of this treatment is that it can prevent premature degradation of the gene therapy once it enters the body, and it can carry various therapeutics. So again, CRISPR, modified genes, any of those. And the benefit of also just making sure that it gets to where it needs to be in the body. So some applications are cancer treatment, so delivering mRNA encoding tumor suppressor genes or siRNA to silence oncogenes, which are basically cancer-inducing genes in the body. Now, a polymer. So this is the only thing in the past couple of gene therapies that is man-made. It is a non-viral vector. So the benefits of it is that it's super customizable. So anything you can imagine being put into a viral vector can be even more diversified by a polymer. And it has a very large payload, so it can be tailored to carry so many different treatments. And it does have a low immunogenicity, so low, it just means low like toxicology and cytotoxicity. And it's also biodegradable, so it prevents toxicity from reaching the patient. Some applications include cystic fibrosis and Parkinson's, two diseases which are very rampant in the US itself. And just an overview of some of those FDA-approved gene therapies and their modalities that I mentioned before, like Becvez, Abecma, all of these are currently FDA-approved gene therapies as of 2024. Obviously, more are being added every day as trials go, and people are finding more things to add to that list. And lastly, we are going to be talking about the ethics of this technology. So, the cost and accessibility. It is $3.5 million for just one dosage of Hemgenex as of 2023. And it is a one-time dosage, but 3.5 million is not accessible for anyone unless they are, of course, a millionaire or a billionaire. And now it is $5,000 just for the production cost of an ex vivo CAR T. So hopefully, as this technology progresses, it does get cheaper and cheaper. And another issue is the germline risk. So, of course, we're worried about off-target modification, where instead of the gene therapy targeting the desired area, it targets something else that we don't want it to. Germline modification. So, what if it passes on to our kids and our future offspring? What happens then? These things aren't really known yet, just because we haven't had this technology for long enough for us to know. And we do have a way to prevent and lower transmission now because of improved safety as time goes. But again, unknown longitudinal effects and human genome enhancement. We obviously don't know what would happen if someone decided to enhance someone beyond their natural abilities. And lastly, some in vivo safety. So the long-term effects of treatment are still unknown, And there hasn't been enough time to really study these effects, but hopefully, as time goes on, we can see how safe they are. And for liquid foam, it is currently used to intensify the potency of a gene therapy while offering optimal safety levels. And it is injected into the desired area to ensure a uniform diaspora of treatment while containing it within the target area, so it makes gene therapy safer. And that's all I have. Thank you.
Thanks so much, Wendy. That was super informative, and I really like to hear it like from Anata's presentation too, because I think like she was doing the mechanisms and you did a lot of the delivery mechanisms. So it was really cool to hear kind of both of those back to back in each other. I really appreciated too that you talked about the ethical considerations about it and the costs associated too, to kind of really understand all sides of the issue. And so I'm curious how you see those costs going down over time. Like what do you think it takes for that to happen and for these to become more accessible?
Right. So in my research, um, I did see that a lot of the portion of the costs are really production costs. So the $5,000 in CAR T, which is a new form of gene therapy, it is bringing down the cost a considerable amount from that 3.5 million. And as people are getting more savvy and making these gene therapies more applicable to a wider array of people, that means that they are becoming cheaper. Obviously, in vivo is still a lot cheaper than ex vivo just because it's kind of like a regular vaccine that you get where you can just um, go to your doctor and get a shot delivery instead of going through a long personalized regimen.
Mhm. Yeah, that makes a ton of sense. Looks like there are a few questions in the Q&A. Let me look. Oh, one is very similar to the question that I asked you about expense, which
Sure.
you just covered really well. And then I see some of your co-presenters also shouting you out for this, but Jen says in the Q&A too that she really likes the design of your slides. Did you design the graphics that are in the slides?
No, I found them. I'm actually kind of some of these squares here, they're supposed to have pictures of um the different types of cells I'm talking about, but I guess they weren't uploaded properly into the slideshow.
No worries. No, but it looked really interesting.
Thank you.
Cool. Thanks so much, Wendy.
Thank you.